this quickly. I need a minute. I cannot share my window. I don't know why. Okay.
Yes, no problem. Take your time.
Okay. Can you see the presentation?
Yes, now it's visible.
Okay.
Dear all, I am Dr. Rajveer Singh, Associate Professor in the Department of Pharmacognosy. Today with us the Dr. Timo Lucas, working as a Forscher in the Max Planck Institute. Topic of talk is tracking the microbes using nanopore sequencing. Dr. Timo, over to you.
Thank you. Although I have to correct you, I'm not a doctor yet. I'm just a PhD student, but I hopefully will be soon. So as just was said, I'm working with nanopore sequencing. So I created a software to help with nanopore sequencing to track microbial communities in a setting in bioreactors. But I will tell you more about this soon. So first about why this project was started. 2019, I had a collaboration with the Environmental Biotechnology Group. And our goal was to understand how caprylate is produced in bioreactors. So then we started a new project after that, and it was supervised by Professor Daniel Dusan. He's my principal investigator, together with Professor Lars Angenand and Ulrike Bierhain, who is also a PhD student. So first about bioreactors. They are artificially built environments carrying out biochemical reactions. So they're kind of like fermenters where you have microbes in it that get feed with substrates and they're producing stuff. So they're used in research, like in our university, where they are built for studying various things, or in industrial application. For example, if you want to treat wastewater, or for production of upgrading waste material in milk production, for example, there we use these bioreactors. They can be very dynamic because we're adding substrates, so stuff is getting in the reactor, and also we are removing things from the reactor, and the researchers might change their experimental conditions while they're performing or while these reactors are running. So they are usually running for a long time, like maybe over the course of a year or longer. And so we have a complex metagenome with many non-culturable microorganisms. So you cannot isolate most of them. Is there a problem with the presentation? No. Okay, sorry. Can you still hear me? Okay, I think so. Sorry, because I got some weird signal right now. So I would just go on. The question that arose then is by whom and what can build certain products in these bioreactors? And the idea back then was to use nanopore sequencing to get more information about the taxonomy and the functional potential of this metagenome. So For example, this is a bioreactor built by Catherine M. Spiritu in 2017. She's now a researcher at the University of Maryland. Yeah, and back then our motivation was motivated by this molecule. It's called caprylase, so it's a carbon chain, a fatty acid with a long carbon chain of 8 carbon, or basically a medium carbon chain fatty acid is what we call them. It is can be used to build other chemicals or as a dietary product. So it's a valuable product that you can sell for real money. And yeah, we wanted to know which microbes can build it and how is it synthesized. And the idea, like I just said, is to analyze the metagenomes for to find some microbes that are capable of caprylate production. So this was the moment that the idea from MMonitor, the software that I developed, was born. So it stands for Metagenome Monitor, basically. So we wanted to take samples. So the researchers, they will take samples out of these bioreactors and then sequence it with the Nanopore sequencer. And then they will have these long read data that gets put into Monitor in my tool, and we will get some results in real time. So while the sequencing is performed, they will get to see the taxonomies and more. Yeah, then they can change their experimental conditions or just wait until the bacteria grow, and some bacteria will die out, some others will grow. And yeah, then by frequently taking these samples, we can do the analysis here and also have a visualization for that. So what Monitor does, it's taxonomic analysis, so meaning what is in the bioreactors, how much of what is the relative abundance of these microbes, and a functional analysis, mainly IQAC pathway analysis, and also things like structural variation calling. So and everything can be done without needing programming, so everybody can do it without a background on bioinformatics or whatever. Yeah, my tool, it consists out of 2 parts. The first part is like the one with which you run the bioinformatics pipelines. Is runnable locally by a very minimalistic GUI. So you just select your analysis you want to run, and then you select your files of where your data is stored. So I will quickly talk about what is done then. So the nanopore reads, they get input for taxonomic analysis either into centrifuge or EMU. These are both taxonomic classifier, with the difference being that centrifuge is used for whole genome sequencing data, and EMU you can use when you want to have 16S rRNA data. So then these tools assign the taxonomy, and it gets put into an SQLite database. So also we have the nanopore reads that get assembled using the fly assembler, yielding us the context, so like longer contiguous sequences. If we're lucky and we have a good sequencing depth, we have then closed chromosomes of these microbes. Metachar gets used to correct the context. We use Diamond and Megan to align it against the NO3 database, and then Megan for binning the context. So finally, we have some long genome sequences from all of these microbes. They are separated by taxonomy, and then we can annotate them with Proca, like it's a tool that annotates proteins out of your genome, so you will know what proteins are they capable of producing. We input this into IQAC Charter. That is a tool that uses the IQAC API to create metabolic maps, and also this gets put into an SQLite database. What I haven't told about is the metadata you can also put into Monitor. So metadata being, for example, production rate of caprylate. That's what I was interested in. That's why we made this possible, or like pH value, temperature, oxygen value, whatever you can imagine. So the second part of the pipeline that I'm currently working on is also enabling structural variant detection and single nucleotide variant detection and methylation detection. These are the pipelines used by Nanopore, Oxford Nanopore, the company that builds these sequencers, and I'm just implementing similar workflows into my tool, so people can also use my tool to do these things. Yeah, so now next I'm going to talk about the second part of the tool. Like after you run the pipelines and your data is analyzed, you can then get some interactive analysis results, different plots to check out your taxonomy, something called a horizon plot, then correlation plots, CAC maps. Yeah, I'm just gonna dive into this now. So this is the first part. Like obviously when you sequence something, you want to know what you sequenced, especially if you sequence a metagenome and you don't really know what's in there. Then you can go to this taxonomy tab. This is just a simple visualization. You can select different plots and export everything. And yeah, like here, there's listed different sequence examples, and you can see the taxonomy is slightly changing for from each day. And yeah, so this is a very basic visualization. What could be more interesting is if you sequence, say, on a daily or weekly basis, then you want to quickly see how is my taxonomy changing. So then I implemented something called a horizon plot. This is a plot that is used in time series data analysis. So we can use it to visualize time series data. And the data is cut into different bands. And we have these different colors, like here red and blue, yellow, here. Depending on how far your values are deviating from the mean, or like from 0, but in this case, the mean value. So what does this mean? The plot gets built when we add new samples. So now after this sample has added, it looks like this. And if we add another sample, it looks like this. And what do these colors mean? Basically, the red here would mean that this Pseudomonas aeruginosa species is decreasing in abundance over time, while, for example, this Streptococcus at the bottom, you can see the abundance is steadily increasing. Yeah, so if you want to know what changed compared to last week, then this might be a good plot to look at. Okay, what is this tool also capable of? We can plot correlations. So some people might be interested in, say, how is my species X involved in like this butyric acid production? Then you would get some different correlation values, either strong correlation, positive or negative, to see, yeah, is there some bacteria involved or correlated positively or negatively to any of my production values or other experimental conditions that we can measure. Yeah, and we implemented different measurements like Pearson, Spearman, or Kendall correlation, and you also will get a like a p-value from a t-test or bootstrapping that will tell you how statistically significant your values are. Okay, and last but not least, we have the CAC metabolic maps. I think most people have seen something like this. This is a map of fatty acid degradation, and these little boxes represent enzymes used for each of these biochemical reactions. They are like facilitating these reactions, and we can see, like in this case, the acetobacter was very prominent and has a lot of these enzymes used for carrying out these reactions. Yeah, that's also what you can get by using this metagenome sequencing approach. So finally, we implemented a little web server. So because the thought was people they're using the lab to sequence their stuff, and it is running for a while, they might go home, and at home they might be wondering what is happening in the lab. So this is not fully implemented yet, but in the next version, I'm still working on this. So the thing is, the sequencer could either be connected to a computer that runs the computational pipelines and the web server, and then we can access the computer locally or remotely. Or what I imagined it to be better is like the sequencer is connected to a computer that performs only the sequencing, and then this uploads the data to an external server. For example, you have only like a small laptop at your lab or not a very strong computer, you could use an external server. Then you upload your data there, and yeah, you can connect to the MMonitor web server with any client you have, like with your laptop or your smartphone. It doesn't really matter. You just call a website and log in, and then you can see your results. Yeah, so plans for future releases would be that's things that was requested from other labs. So implementing a command line version of my pipeline is coming next, and also an alignment approach for taxonomy with a reduced database. This would mean right now I'm using these tools like Centrifuge or Emu, these classifier that are more heuristic based and do not deliver the most optimal solution. So I might also align all these reads and get the taxonomy information like that. It will take much longer time, but my colleagues are working on trying to figure out how to use reduced databases to get alignments quicker. So this is something I'm going to look into. And also we might be able to support short reads or PacBio long reads, not only these Nanopore reads, because there's more sequencing devices used than just Nanopore devices. Okay, and finally, because my plan was to make everything exportable, I just wanted to show what kind of things would be able to be done with these kind of data we get. So, for example, here I used some hidden Markov models to search for genes involved in reverse beta oxidation. On the left, you can see this, and on the right side, fatty acid biosynthesis. So this is just an absence-presence plot. The black boxes mean that a certain bacterium has these enzymes or these proteins in their genomes. A white box would mean it does not have it, and a P would mean, because we also use proteomics for this, that we are certain that this was also expressed as protein, not only found in the genome. Yeah, this is not done by my tool, but this easily, you can just export all the data and then you can do kind of these kind of plots. And you can even find the code for this online, so it's pretty easy to replicate this. And another thing that I did once is building a big tree like this. I think this was created with ITOL, interactive tree of life builder. And I could do this because we had 16S sequences that were predicted from the metagenomes. We zoom in, we can just see these are different. Each of these taxa are basically a 16S sequence in the metagenome, and you can see how these basically the 16S phylogeny, how these bacteria are related to each other. Okay, you can find the code for my software on GitHub. First release was done last November. I'm working on the first full release right now, so you might want to check that out if you're interested in using the tool. And yeah, and finally, I'm gonna talk quickly about recent developments. So I introduced her already, Ulrike. She built this bioreactor. It's a much smaller system. Her PhD project involves trying to figure out syntrophic effects of bacteria. So, in these chambers, there's bacteria, there's a cathode and an anode, and there's two bacteria mainly that we're interested in that can only live in syntrophy with each other, because one of these bacteria is removing hydrogen from this system, and the other would die if it weren't for this one. And we were trying to remove one of these two bugs and see if the other can live if we artificially change the environment by adding or removing hydrogen. So it's just a quick example. And you can see this is a taxonomy plot used or done by MMonitor. This was after inoculation, like it was done a week ago or 2 weeks ago, actually. This bioreactor was inoculated with human feces, and you can kind of nicely see this is only this is 1 day, and 2 days later this is another day later, and 1 more day later, how the for example, this Streptococcus salivarius, it is not much present at first, but then it grows quickly and makes up a huge part of this taxonomy, goes back, so basically it gets reduced again and it grows again. So it's a very dynamic system, and it might actually be useful to quickly see how your taxonomy changes. So because yeah, this is just in a couple of days so far. Yeah, and for a conclusion, this tool helped us in tracking and analyzing metagenomes. It helped finding species involved in caprylate production, and also tracking microbes of other bioreactors. But of course, this is not limited to bioreactors. This is just the use case we had, but you could also use this for like monitoring your drinking water, for pathogen detection, or whatever any metagenome basically. yeah, so I want to thank everybody that helped me building this, and everyone from my group, the Algorithms and Bioinformatics group, as well external collaborators and bachelor students that worked with me, and the CMFI for funding this project. Yeah, so thanks for listening, and if you're having questions, I'm happy to answer them.
Wonderful talk, Timo. Now session is open for the discussion. If anyone has any inquiry, go. I would like to thanks Mr. Timo Lucas for the wonderful presentation. I wish you the very best for the future. Thank you.
Yeah, thank you for having me.
Thank you, sir.